Thank you very much for having me, guys, and thank you for the introduction. Nice to meet you. My name is Bohdan Khomtchouk. I'm here to tell you about this really cool, awesome project that I've been working on over the last several months. And making some great progress on it. It's actually something that I started working on even before I came to Stanford. Kind of something I wanted to do during my postdoc, so kind of laying the groundwork for it, and assembling a great team. So Sable Jonkin, my partner in crime on this, my good friend and collaborator, has been an extraordinary person to work with. So he is the author of the CL-NLP library, which is the main natural language processing library in Common Lisp. So he's the guy who wrote that entire library. So a very knowledgeable guy, and this work would not have been possible without him. And in fact, any great software is never lone person effort. So we always need to give credit to the great people that we work with that make this possible. And Dr. Bohdan, my postdoctoral advisor, has also been instrumental in kind of setting the vision for what to focus in on as useful, and kind of driving the inspiration behind doing some of the really cool algorithms and data structures work that went into this project. So without further ado, let's jump right into it. So this is already slide 2, and I kind of want to test the waters here, try a live demo, just because I'm the spirit of living dangerously and seeing if anything fails. This is a great opportunity for me to show immediately what the software is about and why it's so cool. So let me go ahead and start this up. None of this is prepared, so this really is living dangerously. Go ahead and pop up a browser. So the URL is biochat.ai. See, and already, as you can see, that it fails here. This is an engineering problem. Happens. It's a load balancing thing. That's why I wanted to run it on my machine. Let me try the other Firefox browser. If it doesn't work, we'll skip to the next slide because I have a few things I wanted to show. Yep, there we go. Okay, so Firefox is good. So this is basically the interface. And I'm not going to go into the details of it yet because I want to kind of start with a reference point of what to kind of expect before you even come here. So I suppose many of you have heard of the gene expression omnibus. It's kind of a very popular place to deposit your biological data when you publish a paper. So typically, you would read a paper in PubMed. Usually, if you're really excited about that paper, especially if you're a bioinformatician, you tend to look in like the data availability section of the paper, and it usually has a URL of where the data has been deposited and kind of where you can find it in case you want to rerun some of the analysis, or kind of see if you can integrate that data set with perhaps some other data set that you're currently working on, and see if you can get some integrated bioinformatics synergy going. In any case, usually most of the time, at least from most of the papers I've seen, gene expression omnibus is a really popular place to put your data. And they basically list it out as a URL, and each URL has a unique extension identifier, like for example, GDS10 or GSC 390 something, whatever. It's like an ID that actually links out to the respective data behind the paper. So you would go to the home page of the gene expression omnibus, and then you would put in like whatever is that ID, like for example, GDS 10. Let's say you read a paper, and at the end of that paper was a data availability section, and that paragraph had something about, data is deposited in Geo under accession identifier something. And then let's say it was GDS 10, so you go to the gene expression omnibus, you type it in, you press search, and this is what you get. And this is basically hopefully everyone can see. I don't know how to zoom here. Let's see. Nice. Okay. So usually it gives you like a title of what the respective data set is titled. It gives you a summary of the data set. Gives you the organism. Gives you what sequencing platform is run on, the respective paper that it came from, the citation, as well as download files if you want to download some of the stuff, etc. But the thing was that I was reading a lot of papers, and I wanted to kind of use some cool integrated bioinformatics work. I was pulling this data down, and I was analyzing it myself, and in light of like other data sets that I was working with. And one thing that I really wanted was I really wanted to be able to search for something like GDS10 and be able to find similar data sets to GDS10. So it's kind of like a recommender system almost, you know, like if you search for GDS10, you might also be interested in, let's say, X. Except instead of a recommender system, which would take time to learn my taste and preferences and interests over multiple searches, I wanted something immediate. So I wanted to, like, for example, type in GDS10 and then be, let's say, recommended like a list of top 10 hits that are similar in kind of composition to whatever GDS10 is about. Whether, you know, the summary is very similar, like for example, if it's based on the same biological theme or on the same biological sub-theme, or like the topic is the same, like here clearly we have type 1 diabetes that we're talking about. Like I wanted to find a tool that's able to give me that information and give it to me in a way that is immediate without having to, like, you know, learn my taste and preferences like what you would expect from a recommender system. So the thing is that I couldn't find such a tool, so I decided to make one myself because I thought that this was something that, you know, people might actually be interested in doing themselves as well. Because, you know, the thing is, data does not exist in isolation. Like this specific study does not exist in isolation. It's probably relevant to many other studies that are currently out there, and I want to be able to kind of get that sort of insight in a data-driven way. So that's where I created this program called Biochats. And with Biochats, basically, what you have is you have the same kind of thing where you would type in like GDS10. Oops, GDS10. And you would pick a similarity method, and we're going to go into detail in this talk what similarity methods are. Basically, these are natural language processing algorithms that you can select from. And then you have some filters here that you can select for, like for instance, if you want to filter data based only on the same organism or the same sequencing type or perhaps the same epigenetic factor, you can do that. And you can filter by specific organisms, or you can filter by specific library strategies. And again, this is coming currently from the Gene Expression Omnibus. So basically how it works is you just press search, and here is basically your requested record here. This is what we were just looking at, including all of the metadata associated with it, like the sequencing platform, the publication that it came from, the sequencing type, etc. And then all the closest records that it was able to calculate using, in this case, term frequency and document frequency algorithm from NLP. So what it currently did was it took all of the metadata that is located in GDS10, so specifically these entries here, and it tried to find similar data based on that algorithm. Now what will happen is, in this case I returned 10 top results. You can obviously select how many results you want to return, but you can cycle through the different algorithms. So like for instance, if you want to try the DM25 similarity method, which is basically just a variant of TF-IDF, you press search, and these things get updated. Let me actually go ahead and show you this in real time. So let me go ahead and select cosine similarity here, and watch how the closest records here get updated. Yep, there you go. So you basically have to cycle through the different types of similarity methods, see kind of, you know, what data is coming up, because as a biologist coming in with a specific set of, you know, interests and expertise, some of these samples will make a lot of sense and others will not. And you kind of want to see like what makes sense and what doesn't, and perhaps if something is really, really interesting to you, you might click on it. And that event in and of itself was a very critical piece of Biochat. In fact, if you click on any of these PMIDs that are hyperlinked, a really cool event happens. A block on a blockchain gets created, specifically an event that registers your IP address, your timestamp, your search query, so in this case GDS10, your NLP settings, so for example what similarity method you used, if you used any filters, if you searched in specific organisms, or if you picked a specific set of library strategies. And basically all of that information gets bundled up together, and it gets registered as an event on a public ledger called the blockchain. So a blockchain is basically a public ledger that basically shows everyone's clicks from every single angle. And why this is really useful is because you're able to then, over time, see if there's consensus between clicks. So for example, you're able to see if from very diverse different geographical locations throughout the world, people are making similar searches and clicking on similar outputs. Because depending on that event, you might want to actually reorder some of these results. So for example, if number 5 is super popular for this input, like if it's getting a lot of clicks on the PMID, it might float up to like number 3 or like number 2. So it's collective intelligence driven, meaning that people who are actually using the system are actually kind of donating their knowledge, their expertise, their insight into what constitutes similar data. So it's no longer just a set of natural language processing algorithms. It's also a set of collective intelligence which refines the machine algorithms with human touch, which is so cool because you're able to basically add a layer of, that's human on top of what is basically a machine layer. And then there's another layer on top of this, which is actually even cooler than that. It's actually with respect to citations. So you also take literature citations into account. If, let's say, the paper, the GDS10 paper, so this Eves et al. From 2002, if it cites, let's say, one of these other papers where they cite it, like it'll be something that'll rank higher in the system because you clearly have a citation, co-citation dependence, and you can model that in terms of basically either a graph database or a really nice network. So that basically concludes sort of a very quick demo of what Biochat is about and what it does. And for the rest of this talk, we're going to basically go into the details of how all of that works and kind of the algorithmic excitement behind all of that. So I survived the live demo, almost, by the skin of my teeth. So contents of this talk. So the first thing is the machine, right? The NLP algorithms, the stuff that is able to take the metadata behind your data set and kind of cluster it with respect to other similar data sets in existence. To give you kind of a point of reference for this, if you guys have ever built heat maps before, you know how you can cluster numbers based on like various kinds of hierarchical clustering algorithms like Euclidean distance-based clustering, stuff like this. Same exact thing here, except instead of clustering numbers, we're clustering words. And we're clustering words not just by themselves, we're clustering fields of basically corpuses of little words. And that's the coolest thing about it is that we're kind of expanding that concept. And this actually comes from prior work that I did. A lot of my previous publications were in the heat map field, and I was always very interested in that aspect, especially in the clustering aspect. So this is kind of a great opportunity to take what I've learned from there and apply it to words instead of numbers, which is pretty neat. So that's the machine part of the talk. Then, and by the way, that's going to be most of the talk because a lot of our people here today are computer science and NLP folks, so I've really kind of structured the talk to focus on point 1. However, of course, point 2, the community, as I explained with user clicks, we are recording those events, we're making them accessible through a public ledger, we're able to see them evolve over time, which is all really, really cool. And then the literature, is basically prompting us to say, okay, we know from like the Google Scholar API what work cites what work over the last gajillion years. Can we leverage some of that in terms of a citation network to also as an additional layer train the system? Now, if there are any neural network people in here, this might actually sound like a really cool kind of 3-layer neural network here architecture. And this is something that we're really thinking about right now. We're actually exploring the possibility of using Bayesian neural networks right now, which kind of extend standard neural networks with posterior inference. So Bayesian neural networks essentially combine, if you will, probabilistic models with neural networks in and of themselves. And here we're interested in them because user clicks evolve over time. So we're interested in questions like, how do these clicks re-rank the search output for any given input? Or like, for example, with each new click, each user brings their unique set of skills and expertise in biology to the Biochat framework on a daily basis, right? We know this. And it's a very diverse community. So like your typical fly geneticist would have a very, very different set of expertise than a bacteriologist, for instance. And kind of having such a diverse set of people come in and use Biochat is a really, really cool set of training data that we can collect. And that's kind of the excitement for us. So being able to track user behavior and interacting with the NLP organized data via the mouse clicks that record, as I said, the user's IP address, the timestamp, the input query, the output click down, and the NLP settings directly onto the blockchain. So again, like big picture here, we're refining the machine-generated output results with human touch. And then we're using literature co-citation information to further refine the accuracy of this NLP plus split system, assuming, of course, that the papers that cite one another presumably contain biological data that is related by some common theme or subject. We're not talking about frivolous citations. There's quite a bit of frivolous citations as well, like like either self-citations or friends citing friends, or you submit a paper to a journal like, yeah, you should cite this paper, So we're not looking at that because, like, first of all, that's a very hard problem, and secondly because in the grand scheme of things, that's just noise that ultimately gets drowned out. When you're talking about tens of thousands of papers. So we're not really worried about that too much. So that's kind of like the big picture of what this talk is all about. And now we're going to kind of drill down into the details of the next steps. So what's really interesting about this is how cool is it that you can actually, who knew that you can combine a data-driven study with an actual testable hypothesis. Usually you would think, there are two types of studies in this world, at least according to some people I've talked to at the NIH. There's the hypothesis-driven studies, and then there's the data-driven studies. And here you're actually combining the two. You actually have a data-driven study with a testable hypothesis that you can actually collect data for through a blockchain and actually answer this very interesting statement here, which is that collective intelligence, again, as quantified by observing and learning from multiple users' behavior interactions with the metadata recorded on the blockchain, in combination, of course, with training sources such as literature citations and NLP techniques, is better and more effective in finding natural groupings and emergent structure within very large volumes of biological data compared to arbitrarily depositing this data, just wishy-washy anywhere in an ad hoc manner in some centralized database. So this is actually, this is going to be a very cool thing to see how it pans out over the years. And it's kind of like, in my head, it's a no-brainer. I think this is going to be a very interesting hypothesis to work towards. But at the same time, still, it's going to lead to some very interesting answers and maybe some detours along the way. We will have to see. The thing is that currently, the culture in biology, at least from my experience, is that people usually, when they publish a paper, in the field of biology or medicine in general, they generally have to deposit the data in an open source public repository, either because their grant requires it, like whatever their funding agency stipulates, it has to be done, or the journal requires it. So either the journal or the grant source. And that data, usually you satisfy that tick box, you check the tick box, so, okay, I've deposited the data, and usually it goes into some popular database like the Gene Expression Omnibus. And then no one ever touches it again, okay? And that's a really sad state of affairs, because you have accumulated over the many, many years such an extreme wealth of data that all of a sudden you don't have the infrastructure to really leverage it anymore. You have all these kind of like silos that are used as storage repositories to store the data, but how do you actually reason about that data? How do you compute on it? How do you integrate it? So that's where Biochat came in. And the name Biochat is kind of inspired by allowing the data to chat with each other. So teaching biological data sets to learn to talk to other data sets using, as we mentioned, a combination of machine algorithms plus human touch plus literature citations. So kind of the motivation is that currently there exists, a lot of different databases, thousands in fact, like the Gene Expression Omnibus, Mirbase, TCGA, etc., all containing terabytes of publicly available data. TCGA, for instance, has 2.5 petabytes of publicly available data. Guys, that's a lot of data. And some of the things that we gotta remember is that with so much data scattered in so many centralized locations, it's kind of virtually impossible to facilitate any level of database interoperability at that scale. And that, of course, makes life hard, right? It makes the process of computational unification and kind of driving data-driven biological discovery, it makes it pretty difficult. So you might think this is a new problem. Nope, this is not a new problem. This is actually a very long-standing research question. In fact, the whole research question of making heterogeneous database repositories interoperable is actually a very, very old problem. And it goes, for instance, all the way back in time to, again, my university, 1994. I was 3 years old at that time. And Like, just to kind of give you some insight, and just read like the first paragraph of that abstract, and you will already see that even back then, they had 100 existing molecular biology databases. This was 1994. Okay, and people were already thinking about this. This was, of course, very much ahead of their time. And it still is. I mean, look, I'm working on it. I mean, this is still a very hard problem, and people have proposed various different kinds of solutions. And kind of, I want to show you what I'm trying to do and why I don't necessarily think it's better than what's been done before, but I think it's a very nice complement to what's been done before, and it could actually kind of be that turning point where it actually all of a sudden clicks and everybody's happy at the end of, I don't know, the year, the next 2 years, whenever I'm satisfied and done with this project, then it gets published. So it's also a nasty problem. Look, there's like a website that actually calls it a big, hairy, and nasty problem. It's like a big gorilla, right? It's the 800-pound, gorilla in the room. Making heterogeneous database repositories interoperable is a pretty nasty problem. There's no way to sugarcoat it or make it easy to say. It is what it is. And big gorilla is not even a biological focused database. This is just a general framework that you guys can check out. That basically, just in general, data integration is a problem on a worldwide scale. But we're exclusively focusing all of this research on biological databases. So today, unlike in 1994, biological databases have a very unique set of properties. I would actually describe them as a heavy tail distribution. Why heavy? So here's the thing, back in the 90s and back in the 2000s, huge top 10 databases like the Gene Expression Omnibus that I showed, or TCGA or SRA, etc., they just didn't exist. Today they do, and most biomedical research papers you read in PubMed contain data that's deposited in one of these popular databases. Therefore, these top 10 databases capture essentially what is the lion's share of the data, most of the data. Here they would be represented on kind of the left side of this graph. On the right-hand side of this graph, you would expect to see exotic sort of domain-specific databases, usually catering to a certain research niche, and getting more or less, I would say, the same level of usage relative to each other. Because, again, these are not like kind of like behemoth databases like the Gene Expression Omnibus. So you have a more or less kind of like, constant amount of usage down in tables. Often the data in these exotic databases is actually also present in one of the top 10s. So there's also often a duplicity problem. I mean, that's a whole other can of worms that you have to open. Is that sometimes, and actually, well, very often, the same data set can actually be present in multiple different databases at the same time, which, is all sorts of computer science headaches, if you will. Now, the other thing that's different in 2018 versus 1994, besides for the fact that this is what the state of databases looks like, is that no one's actually surprisingly tried applying blockchain principles to any of the metadata stored in these databases. So all databases have some way to standardize their data, and usually they're not interoperable. And that's a big problem, and that's a problem that people have known about for a long time. At the same time, most of the good databases at least have a requirement that its users have to upload a certain set of metadata describing whatever is the contents of the file, or a set of files that they upload into the system. What if you could go through all these thousands of different databases, or even the top 10? Let's start with the top 10, since we already know from this previous slide that if we capture the top 10, I mean, we're great. This is great, right, if we get the top 10. Grab the top 10, grab all the metadata from the top 10, and then use these kind of blockchain principles to be able to see what can we do to have them start interoperating with each other, sort of in a peer-to-peer sort of way. So what you're effectively doing is you're decentralizing the metadata. So you have centralized storage repositories like Geo or TCGA, and all of a sudden you're decentralizing the metadata behind all the contents of those files, and you're storing it on the blockchain. And then what you're doing is you're interpreting user interactions with this blockchain like as if they were transactions. So blockchains come from a cryptocurrency beginning. And right now, people are getting excited about applying it to many different applications outside of their original intended kind of,
use case, which is to buy yourself a coffee in a Palo Alto cafe. There were bars in Palo Alto back in late 2011, even early 2012, that would actually accept Bitcoin for beer, and that's pretty exciting, I think, especially if they sold at the all-time high, right? Because they were basically selling you a Guinness or an IPA for thousands of dollars essentially. And all that information you can go check it out. It's public ledger, so it's all blockchain, it's all there. You could see those transactions happen. Similarly, many years down the road, like here we were talking about something that was happening 7, 8 years ago, you're still going to be able to see people interacting with data 7 or 8 years ago. Because the thing about blockchains is that it's extremely transparent. It facilitates, a level of, as I said, transparency, security. I mean, different things that we don't necessarily have to worry about as much for something like publicly available data. But as soon as you kind of cross the line into clinical data or like sensitive, like pharmaceutical data, like clinical trials or something like that, then you have to start worrying about later on security. We'll actually talk about that later on in the talk. But just kind of want to foreshadow that a little bit. So again, the significance of the study is that, as mentioned, right, this data is growing every day. Back in '94, we had about 100. Today we have thousands of different databases. The ecosystem looks very different, and there's still new experimental data being published daily. Biology also is done in very many different model organisms. So anything from humans to mice to rats. And it uses a whole slew of different technologies, next generation sequencing technologies like ChIP-seq, RNA-seq, ATAC-seq, microarray technologies, etc. Many different cell lines as well. So if you want to like understand and integrate cell lines, this is actually a really interesting one. So the Broad Institute in October 2017 launched an international consortium effort called the Human Cell Atlas. Whose goal is to actually sequence every single cell in the human body over, I don't remember what exactly the timeline was, but that's 36 trillion cells, guys. 36 trillion cells. Imagine what kind of data explosion we're looking at. I mean, we're talking about pre-Cambrian era right now. Like, when these guys are done with this in like 10, 15, 20 years, this is going to look like easy stuff. And if we don't have the infrastructure that I'm building right now in order to meet that demand when it gets there, it's going to be very, very difficult to cope with that sort of load. So my philosophy with bioinformatics and computational biology in general is to be able to see the ball way before it gets there. Or I think Wayne Gretzky said it with hockey, see the puck before it's even there. Right? You have to see where things are headed and kind of be able to produce the tools, produce the infrastructure, produce the supply to meet people's demand down the road. That's really, really important because otherwise it hinders science, it decelerates the whole process of discovery, and in general people aren't very happy when they don't have the tools to get the job done. Again, also, all people are still investigating many different types of transcription factors in many of these cell lines and different sequencing types. Epigenetic modifications actually come from an epigenetics laboratory. We're actually a cancer biology laboratory specializing in epigenetics. I work on the aging arm of the laboratory, and I'm the data guy, so I do all the bioinformatics analyses as well. And just kind of, I know what people are doing currently and how it's the problem is just getting bigger and bigger and bigger and bigger, and someone has to do something about it. Because multidimensionally integrating all this data is, it's pretty essential to biological discovery because there's many different data sets out there where you would at first glance be like, oh yeah, no, that's cool. But then at the same time you'd be if you really pay attention, you might be wait, this might actually answer some specific facet of the research question I'm addressing, maybe from a slightly different angle. Being able to capture that information from the community through user clicks is really cool. Because it kind of it takes the cognitive cost, the cognitive load off of you, and kind of puts it on the community in general. It distributes it in an interesting way, in a distributed computing way. So it makes the problem scalable, and it makes kind of tackling, the 800-pound gorilla, or the elephant in the room. Someone's got to address the elephant in the room, and that's me currently. I'm trying to address this elephant in the room. I'm trying to get you guys excited about it and see perhaps, if you want to collaborate on this, this is a very big project. There's many different angles you can attack this on. Coolest thing is that, I mean, even years ago, this was the Obama's Cancer Moonshot program. They said, wait a second, what if cancer cures could already exist in big data? So what if all the stuff that we currently already have out there in the Gene Expression Omnibus, in the Cancer Genome Atlas, etc., what if there's ways to actually integrate it and actually find answers to the research questions that we're currently posing in the lab, as opposed to generating new data. Now, there's nothing wrong with generating new data, and in fact, it's a very important thing because a lot of the biological data currently out there is total crap. And like my PI can personally attest to this, like there are very many different research questions that people have addressed over the years using biological techniques and just methods that in general could not be reproduced by other groups. If you can't reproduce someone else's findings, even if those findings are highly cited, a whole kind of like research direction in the field of science goes in another direction. So producing new data is still vital, but producing quality new data is what we need. Now, before we come to that stage, what if we could try to find some of these answers in the currently existing data? So teaching biological databases to learn to talk to each other is kind of like that little chat icon in the middle, that little GIF thing, right? You're kind of allowing an intercommunication to begin between these various different database repositories. And again, you don't have to worry about standardization here. You're just worrying about the metadata and being able to compute on the metadata. So that's the coolest part about it, is that you're allowing data to learn to talk to other data, and that's why this program is called Biochat. And that is the URL, www.biochat.ai, and then the code, which is all written in Common Lisp, is located in the Biochat repository. So now starting to get a little bit into the details. So again, we kind of covered this. This is a web application, right? It runs a variety of machine learning, natural language processing algorithms that ultimately, at the end of the day, they group the biological data records by this or that similarity method based on their free text description and other metadata information. Currently, this data is coming from the Gene Expression Omnibus. I chose the Gene Expression Omnibus as kind of a playground to start this in. It was very popular, in the last many years of PubMed papers, and because it's very big. And it's very well curated as well. Most of the metadata in there is very nice. Of course, in working on Biochat, I found many examples of not so nice to play with data, but it is what it is. Nothing's ever perfect. In fact, metadata is such a hot topic right now in terms of bioinformatics and computational biology that last year somebody published a paper called MAT-SRA in the Bioinformatics Oxford University Press Journal that literally was able to standardize all the metadata in the SRA, the Sequence Read Archive database. Just to kind of give you insight, you can publish big papers just by standardizing metadata because it's such an important problem to address. Because it basically makes data that much more variable. And that's what we're focusing on with Biochat. And the Biochat, by the way, is synced with the Gene Expression Omnibus every 24 hours. So in the back end, I have cron jobs that are basically running around the clock, syncing the data that's coming into Geo with Biochat. So Biochat is always up to date. I don't have to worry about it. I automated it, made my life easier. It works, and I'm happy. So similarity. So similarity is going to be a very, very key kind of concept we're going to discuss for pretty much, well, not the rest of this talk, but a huge part of it. Similarity in the context of data set descriptions, actually, believe it or not, it's not a very well-defined concept. Because, first of all, what if you have something like this, right? You have first of all very many fields. From the point of view of a researcher, the other thing is that you might have different notions of what constitutes similarity. As I showed you, when you loop through the different options in the Biochat UI, you basically cycle through the different similarity methods, and then you look at the output. And the output that you like, you're naturally probably gonna click on it because it's curiosity driven, right? You want to learn more about whatever is the study that produced it. The other thing is sometimes you might want to only focus on a specific or particular group of organisms, whether it's the same organism as your search query, or perhaps 2 or 3 or 4 different kinds. And then also more nuanced cases when only experiments that target a particular epigenetic factor are requested. Or for a group of epigenetic factors, like there are certain papers and certain data sets that address both like H3K9ME1 and H3K4ME2. Like those are two commonly studied, in tandem in publications, chromatin marks. So they're very often examined in studies like that. So that's why Biochat, the project in and of itself, it aims to provide a flexible tool set suitable for experimenting with different kinds of similarity measures and their parameters, as well as kind of supplemental filtering based on those additional parameters, as discussed here. So measuring similarity, this is where we get a little bit more technical. So in the context of Biochat, the similarity measure is basically a function of two records that returns a number in the interval 0 to 1, signifying the degree of similarity. And the closer it is to 1, the more similar it is. Now, the trick is that the magnitude of the similarity does not have any particular meaning. The only requirement is that the records considered more similar should have a much larger value for their similarity. That's why you can't actually compare similarity measures across the different types of algorithms, because all these algorithms are based on different techniques, and it would be, I think, at the end of the day, it's more useful to compare them from a biological standpoint than it is to compare them from a computational standpoint, because we want to capture the actual biology more than we want to capture the machine, right? That's why we added the collective intelligence component. Kind of, getting a little heads up on the NLP algorithms. Currently in Biochat, there's basically two principal approaches that we use. One is a back-and-forth-based similarity, which I'm sure many of you are very well familiar with. The other one, distributed representation-based similarity, which also is a very common technique. But just to kind of quickly go through this, we're not going to focus too much on these next few slides. Just kind of want to get the basics out of the way so that everything else makes sense. So in the back-and-words approach, the token-based approach, each record's textual description is transformed into a sparse vector of the size equal to the size of the vocabulary. What does that mean? That means that basically when you have that record, like GES10, and you have the different metadata fields, you pull out the words from each of those metadata fields, and then you can basically create a sparse matrix out of them, and then you can take that approach, and it's actually, well, it's bread and butter natural language processing stuff. So nothing too out of the ordinary yet. The transformation is performed by tokenization of the text. So you turn the words into tokens and then assign some weight value to the element of the document vector representing each token. The back-and-forth similarity measures include currently variants of TF-IDF, like for example the vanilla implementation of TF-IDF and BM25, which we will actually cover on the next slide. The TF and IDF vocabularies are calculated from the whole record set, so again from every single field in the metadata description, using, once again, tokens from the record's title, summary, and design description. And the TF count for each document is calculated as a ratio of token count by the document's length. So specifically this. So the IDF count is calculated using standard log weighting as so, very simple stuff. Note both the TF-IDF and BM25 similarity vector calculations use the stored weights. That's really important, actually. But I won't go into the details of why at the moment. And you can ask me very much. Also, similarity value of two records is calculated as a ratio. So again, we're talking about trying to look at the L2 norms here. And I have a formula here somewhere. Yeah, there we go. So this is actually the formula that we use. And what's nice about all of this is that we're actually able to leverage a lot of the previous kind of bio-NLP work that was done by some really talented people who basically broke down all the PubMed articles and kind of distilled the word corpuses in the PubMed database into word vectors using word spec, and we kind of followed their approach. So another approach that is used to do this in Biochat is actually based on this. And I just want to pause here for a moment and just give a little hat tip to some of the really cool stuff that was used for constructing vectors using machine learning algorithms, like for example, paragraph vectors and escape thought vectors. So this is all NLP based stuff that was able to make this all possible. And working with Stanford on this, who wrote the main natural language processing library in Common Lisp, he's like an algorithms and data structures expert, and it's just been a real pleasure, to kind of be able to glean some of that insight from him. Like, what should we focus on? Like, where should we put more weight in our data sets, etc. And kind of, he's always asking me questions like, okay, so I don't know anything about biology, but if you can tell me, like, what do biologists really care about? Like, and then he throws like 5 or 6 different data sets at me. And we like go ahead and brainstorm like that, and it's great energy. So I kind of just want to float out some more of this out there. So in Biochat, what we did was we took the lead of the guys who created the PubMed vectors, and we used Word2Vec. So the reason is threefold. One is availability of high quality pre-trained vectors, again from the team that already did this. Also lack of training data for the successful application of doc-to-back approaches. You'd be surprised not that much has been done on that. And in fact, bioinformatics is kind of the Wild West, guys. There's so much great stuff that you can do, like as a dissertation topic or a master's thesis. Like it's so many low-hanging fruit that you can knock out quickly. It's phenomenal. And third reason is empirical results showing that simple word vectors aggregation actually performs no worse than a short text. So some of those metadata fields are very short. So like for example, like organism, it's always going to be either Homo sapiens or Mus musculus or Ratus rubescens, right? These are very short descriptions. But believe it or not, that doesn't really matter. So Word2Vec is able to handle that, and there's ways to work around it in very elegant, nice ways. So kind of using Word2Vec in Biochat, here we're talking about basically matching words by similarity using vector space representations. So as you guys already know, vector space models, they represent or embed words in continuous vector space where semantically similar words are mapped to nearby points. So they're kind of embedded nearby each other. Word2Vec is particularly computationally efficient for this matter. So learning word embeddings from raw text is really easy to do with Word2Vec, and each record is basically transformed into a vector, and that's basically how we get the job done. And again, all of these vectors are PubMed vectors. And PubMed is, by the way, I know I haven't clarified this at the very beginning, but PubMed is the primary biomedical literature database. So if only we had something like that for data, but we don't, right? There's thousands of different databases. But for literature, it's easy, right? Everything that ever possibly came out in the biology, medicine, biomedical space is most likely in PubMed. And sure, there's many preprints out there. There's many IEE journals that will not appear, but at the same time, like the lion's share of the literature is there, and that's useful. So the choose similarity methods option, the one where I showed you how you can cycle through the different types of similarity methods, then rerun it, and then examine the output and see what clicks. There's actually some really cool stuff going on here as well. So you can do the comparison based on, and we saw this already. We can do cosine similarity. We can do smooth cosine similarity. Again, these are mostly details that we don't have to worry about right now. But the point is that all this stuff actually comes from 2 things. One is understanding the NLP algorithms behind this, and 2, kind of leveraging the biology, and actually 3, also leveraging the work of these people who created these kind of pre-trained word vectors from the PubMed corpus. Those things come in very handy. And since the main application of Biochat is sorting the georecords, it's very useful to have a filter, right? If you only want to retain records that are for a specific organism or group of organisms, or only retain records for a particular histone or set of histones, it's very, very useful. Now, the recommended usage, as you saw from my live demo, is cycling through all the available similarity methods and examining the output. And then the question is, can we record which output looks good and which output looks bad for each NLP algorithm and input query? So when I was first, again, now you guys know like how I do that through clicks. But before I came up with the clicks idea, at first I thought, this is kind of an interesting historical point, I thought about having like a YouTube style, thumbs up, thumbs down. And I was like, really excited about the idea. I'm like, it's gonna be really cool. It's gonna be the next YouTube. And I was like, no, because it's gonna be very onerous on the user, right? It actually asks the user to do something, as opposed to have natural curiosity drive them to make the click, which is really the same exact thing. So it's onerous on the user. And it's really a burden when presented all at once, because all of a sudden you have more than a dozen pairs of like thumbs up, thumbs down signs looking at you all on one page. So it really significantly clutters the UI. And I'm really passionate about design. Like design matters a lot to me, and I think maybe that's because, growing up, like I was still the generation of people who remember the 30 gig iPod and how sleekly designed that thing was. And then from an engineering perspective, I think it was a marvel. But like even as a young kid growing up, like design like really influenced how I think about software. So to me, when I think of a UI, it has to be well designed. It has to be, it has to be fun to use. The user experience, the user interface, the UX UI has to be engaging. And that's why, ultimately, I started asking the harder questions. And I didn't want to clutter the UI. I didn't want to have the users feel like I'm asking something from them. So I basically decided, at the end of the day, that I was going to use a more clever method. And that is basically the hyperlinks method, where the clicks are curiosity driven. Now, if you're really interested, again, in one of those outputs, for example, because it looks so relevant to your input, like, number 6 or 7 or 8, like let's say number 7 looks extremely relevant to your input, I mean, you're very likely to click on that PMID, that PubMed ID, that's what PMID stands for. To navigate to the respective publication to, well, essentially learn more information about the respective study that generated the data. So in doing so, your click is registered as a new block on the blockchain, and where each block, as I mentioned before, it contains the user's IP address, timestamp, input query, output cookdown, NLP settings, all that information. So actually, we're going to cover that a little bit more in detail on the next slide. But for now, the technical message is, in this framework, user interactions are treated like as if they were transactions. And you can, similarly to transactions, like when you go on your banking history or when you go on Coinbase and see how, like what you've been purchasing and stuff and taking out and putting in, you can track the provenance and monitor in real time on the blockchain. So you can always track the provenance and real-time evolution of the system as people use it. And that's really exciting. Especially, looking back on it, I think 5 to 6 years from now, we'll see kind of what people are searching, what's hot, what's trending, right? You can like capture all that information. There might be a paper 10 years from now just on that alone, just to kind of monitor, the trends of how biomedical interest has evolved over the day. I mean, that's pretty cool. And you can capture all that information publicly through a public venture, through the blockchain. Very cool stuff. So you're treating these interactions like they were transactions, which is you're going to be monitored, creates new blocks with all of this information. And kind of moving forward, similarly to how blockchains work, you need to establish consensus. So this is similar to the concept behind the proof of work algorithm. The ranging and sorting of the output results will only be effective system-wide once consensus is reached amongst a sufficiently diverse set of users. Again, similar to the concept of cryptocurrency miners competing against each other to complete transactions on the network and getting rewarded for Bitcoin. Here it's actually a little different. So instead of being offered a coin as a reward for your mining efforts, users are actually rewarded with a rewarded set of output results that more accurately reflect current community consensus. No, current community consensus. The consensus might change later on, but it reflects current community consensus for top-ranked hits via the collective intelligence that was captured during the onboarding process. So having users come in, get onboarded, click through this, and then establish some consensus across a sufficiently diverse set of IP addresses geographically worldwide. So that's very useful because you can't actually, I mean, I've thought about creating like a coin out of this just for fun to see if I could motivate people like through finance because, academic funding is running dry and it would be really nice to have an alternative source of funding besides for just governmental funding, like cryptocurrency could potentially be something that is a viable alternative source of academic funding. Could be, I've thought about it. The problem, at least in this framework, is that you could game the system. you can cheat on it. So you could call up, your ex-buddies from X number of places around the world. You could be like, hey buddy, can you perform this query and click on that output, and then I'll log in 5 minutes later and do the same thing, and you do that too, and you could game the system. There's actually ways around it. I've actually thought of ways to work around that, but it's currently not something that is very high priority on my list. But again, all these scenarios are very comical if you're not talking about money. I mean, if this is altruism driven, if this is curiosity driven because you want to click. Altruism driven because as you use Biochat, you're actually donating your expertise to the system. So you're actually kind of imbibing instilling the knowledge that you have of your specific field area of expertise, like as a fly geneticist or as a bacteriologist, into the system itself. And the AI is able to soak in that information like a sponge. So therefore, Biochat is only currently driven by altruism and curiosity, not greed, not financial greed, although there are ways around it. Like for example, you can use Byzantine fault tolerance to fight people who want to game the system. It is possible. I'll actually mention that on the next slide, on the security slide. But again, currently not something that we need to worry about. So different incentives for different folks, right? For any given input query, users are motivated and incentivized just by curiosity alone to click on the PMID outputs. This activity may or may not match the double-blind activity of other geographical users. The reward instead of a coin is a rewarded set of output results that reflects that community consensus. And that kind of, coining a new term here, right? Coins, because, it's kind of fun word choice. No one was really doing anything like this when I started, and I tried describing this concept to people, and they kept asking me to just summarize it in a punchline. I'm from Stanford, and people there are all about, elevator pitches, and, my time is limited, so, pitch it to me in 10 seconds or get out. And that kind of, made me realize that, if people kind of want to, have that limited way, constrained blinders on way of thinking, maybe it's time to actually call it something. So I decided to call it the Internet of Omics, because that's what you're talking about here. You're talking about if you will, you guys have heard of the Internet of Things, right? Where you have, different hardware devices talking to each other. You could have your coffee machine make you an espresso using your phone, right? You can actually, if your phone is hooked up to your coffee machine, you could, press a button and it'll make a coffee for you. And thanks. And then you drink your coffee instead of going to the kitchen and doing it manually. So it's kind of similar in that sense. That you're allowing now, instead of hardware devices communicating with each other like that, you're abstracting the concept of hardware to include software. So you're actually allowing data to talk to each other. Now the thing is that the Internet of Data, when people use the term Internet of Data, they're usually referring to kind of the logs that get produced from your phone sending a query over as a request to your coffee machine. So it's a totally different subject, very fascinating. It's all like XML and stuff. But here, we're not talking about the Internet of Data in that sense. So I realized we needed a new term for it. That's the new term. If this thing gets big and is a big publication and ultimately everybody uses, then you'll be hearing that a lot. And this is kind of the first public venue where I've disclosed it. Internet of Omics, there you go. It is what it is. I needed something to refer to it both locally at Stanford and amongst my colleagues. So this is a very cool thing. I was actually waiting to get on this slide, and I was chatting to some people today about it. What if, what if someone you really didn't like in high school, or someone didn't really like you in high school or college, and it was like, oh man, if I could just go on to Biochat and unleash this malicious spam bot, or just click on random PMIDs and get the system out of whack. How robust would Biochat be to an attack like that? I mean, would it be able to withstand a coordinated, serious, professional, non-script KD botnet attack? That's a really good question. So I actually am currently doing some of this stuff, and no, so far, and I've been teaming up with some people at Stanford for some security guys. They've been, we spun up like a local instance of it, tried hitting it with all sorts of stuff. And what's interesting is that good luck. Here's why. So the literature citations is a really, really good shield. Usually when someone unleashes a botnet attack, it's not smart enough to know biology, especially at a global scale. So it's going to make more or less random clicks, and random clicks will not match up with citations in any way, shape, or form. So now what we did was we tried to train these spam bots to actually think like biologists and train them on the citations to actually go the other way around. And again, it doesn't work because with blockchains, everything's fully transparent and you can shut down IPs as soon as you see a flurry of activity that is totally like random and that's not matching the query. So you can't actually spam Biochat, which is really cool. And then, of course, as I briefly mentioned, Byzantine fault tolerance is another future direction that we're currently considering. Personally, I find security to be very fascinating, and it's not very often that you get a chance in bioinformatics and computational biology to really think of a white hat, black hat hacker kind of, dual, dogfighting, like two fighter jets with each other sort of a thing. And that's just, I'm having some fun with this right now and kind of seeing what I can do with it. It's a pretty fascinating field. In general, cybersecurity is a fascinating field. So, the take-home message of this talk, decentralized. Metadata, extremely important, has to be high quality. Like next time you publish a paper or your friend publishes a paper, send them a thank you note if they do a good job with being very descriptive in their metadata, because ultimately Biochat is nothing without the metadata, and the metadata is what makes this entire system come alive. And basically, being able to compute on these metadata descriptions through a combination of human-based and machine learning-based approaches is just, I think it's the future of where we need to go with this field in order to kind of tame, the dragon of the biological data deluge that we see at the moment. And some future directions of where we're currently taking this. So we want to do some really cool GPU-enabled stuff. Because this is a very computationally complex thing, you're putting in a query, and then you're basically running a huge sparse matrix. And this could be hundreds of thousands of rows by hundreds of thousands of columns. Unless it's pre-cached, it's going to be really hard to get good performance. And that's a big deal because if people are going to cycle through the different options, they're not going to want to wait for 2 or 3 minutes, which is how long it currently takes. Like the results that I showed you earlier today, those results were all pre-cached because I had run that query before when I was showing like my lab and stuff. But if you were to type in something that hadn't been typed in before, it wouldn't be pre-cached. It would take about 2 to 3 minutes to run, which I have like a little spinning ball. But at some point, people are going to like start posting this on Twitter and be like, spinning ball of Biochat death. I really don't want to be that guy. So what I've decided was that I'm going to make my code base run on the server side using GPU-enabled clusters. Specifically, I'm going to pre-compute all of the different pairings on Sherlock. That's Stanford's high-performance computing cluster. And then I'm going to store cache the results on Amazon Web Services for basically easy fetch and retrieve. And that's going to be really, really easy to do because it's only about 20 gigs of data in general, text data, and that's nothing to fetch and retrieve. So I've already kind of done all the back-of-the-envelope napkin calculations for doing this, and kind of got the A-OK from the Sherlock team. And then any new metadata records that come in within the 24 hours are all daemonized. So basically within 24 hours, they get like 10, 15 new data sets. You can get that done within 24 hours. So it's really nice because you get to experience the sheer power of the GPU. You get to daemonize your code. I mean, it's pretty great, fun all around. And of course, continue adding new metadata records from existing biological databases. As you know, currently Biochat is only the gene expression omnibus, and removing any duplicate records as they come up. And in general, yeah, keep growing Biochat and making data social, if you will. If you want to kind of put that into 3 words.
To acknowledge my funding source here from the NIA, and then the computing that I'm going to be doing on the Sherlock computing cluster. And yeah, that wraps it up. Q&A. Thanks a lot, guys.